So this afternoon, it's going to be very innovative, very exciting. You're going to hear a lot of rising stars talking about their latest cutting-edge research and also their visions of how education should be in the future. So by the first part, let's talk about research grant, the most exciting part. This is going to be a perfect year for people to apply for grants. So if you're interested, please try this year to submit your grant. For our grants, what differentiates us apart is that we put special emphasis on innovation and promoting junior faculty and to communicate the bridge between research and industry. Our grant has 4 unique components that I think is well established. First, and most importantly, there's no geographical regional restrictions. Anyone interested in research, eligible for research, can apply from anywhere in the world. It doesn't matter where you're from. Actually, we encourage people from different regions where there are deficient in grants to apply for our grants, because our goal is global outreach research for everyone. The second part is the budget aspect. It's very exciting that thanks to our chair, and they decided, Dr. Dai and Mr. Dai Feng and Donald Huang, and this year, they generously allow us to have up to $700,000 funding. So we have sufficient funding for you to apply, and this is the biggest, I believe, international research funding in the world. Third component is crucial too, is that this is going to be a very fair and comprehensive evaluation process. So do not worry about, I don't know anyone in the committee, what should I do? No, this is going to be a fair and going to be a very comprehensive evaluations. We'll look at your application carefully in detail to make sure every application get the proper evaluation. And we'll give you comments in the end. Even you get a grant or not this year, this will serve as a base for your future application. And we always encourage a resubmission grant. If your grant is tagged behind under the score line, we encourage you to use the comment to reapply for the following year, okay? And we have a very good reviewers. They're experienced. And my co-host, my co-chair, Dr. Ko, is with me here. And we have a array of panels of committee members who are experts in different fields in orthodontics, AI, genetics, biomaterial, in every aspect. So we'll make sure that your application get properly reviewed. Let's look last year. It's our first year with this grant. And what we have is that we have around over 64 applications, 34 projects, and we funded 16 projects. We have invited 13 reviewers from all over the world to give the review. And we look at the breakdown of numbers, people who got funded. We got Asia, North America, and Europe. And what we really, really would like to see is Dr. Harvey here. We would like to see people applying from different regions, from South America, from India. We do have application from India, and the application hopefully from Middle East, so that we want to make sure truly this grant proposal it's going to reach out globally compared with other foundations. And this is the breakdown of how many people, what they have got. So we have primarily faculty, but we would like to encourage the young people, because young people are the ones with really good innovative idea to apply, okay? And we do have a comprehensive program to encourage and to mentor young people. We don't just give out the money. We help the mentorship. We give advice. We help the review papers. With your first submission for other grants, we help all this process. Oops, sorry. This shows you the people who got the grant from last year. And as you can see, as I mentioned again, people from Europe, from US, and people from Asia got a grant. But as I mentioned again, we really want to reach out to more people in more countries to apply for the grants. And again, if we find grants that's worth it, if we reach our budget limit, we always talk to Dai and talk to Donald, give us more money, because we want to fund people. And it's always good to have people who support us. And this is going to be the review panel for the coming year. We further strengthen the people here. We have 19 orthodontic experts from 7 different countries. As you can see, the new member we add, Dr. Fleming from Ireland, Dr. Nanhash, Michigan, Dr. Chijiro Tanikawa from Osaka, Dr. Zhang Wang from Sichuan, Dr. Guang from UPenn, and Dr. Ren from Europe, Netherlands. So we have, and of course, Dr. Fimon from University of Illinois, in addition to our current members. So we make sure that application will get a fair chance to be evaluated. And what type of grant do we have? We have 3 categories of grant: young, elite, and clinical grants as Dr. Wu mentioned, to cover from young researcher all the way to grow up to the career to the top. So we have different layers of grants. Young research grant, the most exciting grant also, I think. We provide up to $15,000 for up to 2 years for renew applications. We open for graduations, for graduate students, all the way up to assistant professors. Depends on which level you feel you are comfortable with. And the elite grant is $30,000 per year and up for application for 1 year renewal. And this grant is designed for assistant professor up to full professor. This funding will give you for any ideas and for junior grant, any idea you have, but you don't think it's quite ready yet for a major government grant or industrial collaborations. So we'll give you enough funding and support so that you have enough data to apply for a much bigger grant, a much bigger collaboration. Clinical research grant is open up for multi-center between universities. As you know, this year we have multi-center grant awarded to 2 different countries, US and China, as a collaboration. That's what we would like to see. Hopefully we can get collaborative grants from between Middle East, South America, and China, and US and Canada. So hopefully this will, and by the way, Carlos, Canada is not part of the US, I know. So this year, this is the time we just formally start our application, as if today, yesterday and today. So please go online and look at the applications, and please, for faculty, for senior faculty, go and meet with your junior faculty and your other faculty and help them to work on this grant. But remember one thing, any questions you have with our whole team here, I think you all know probably, you have Angela, you have HD, you have Vincent, you have Wells. They are all part of our administrative staff. They'll be here 24/7, answer any questions you have on this grant. So we want to make sure you have a very smooth application process. What are the areas that we want to fund? As we mentioned, we want to fund innovative area, translational area on the research, okay? These are the topics that we suggest, but not limit to these topics. Any exciting thing out of the box thinking, please submit to us. We will always review them. And finally, this is our QR code. Any questions, feel free to ask us. As we mentioned before, this is the Chinese saying we put it up here. I think it's very true that it takes decades to grow a tree, but it takes centuries to cultivate talents. And it's our responsibility for the next decades and the future to really grow the rising star here. So IOF, we are committed to help the future of our teachers and the leaders of our professions. Thank you very much.
Thank you very much, especially for me. I'd like to express my thanks to IOF Foundation to support the research, the clinical research of our team. Thank you so much.
We're excited to see your data from hopefully new incidents, new discovery on face mask expansion, all the new technology you have. Hopefully we can use on clinical setting very soon. And next, our staff spend a lot of time to make a very nice video to summarize, and they insist they have to play it for you. So please let's show the video.
Okay, thank you.
Then International Orthodontic Foundation's research grant video presentation. As a nonprofit organization, our mission is to promote and advance orthodontic education and research around the world. In this tutorial, we will provide an overview of our grant application process and guidelines to help you submit a successful proposal. Our foundation supports innovative research projects that have the potential to make a significant impact on the field of orthodontics. We welcome proposals from researchers around the world who share our passion for advancing orthodontic education and research. By choosing to apply to the IOF Research Grants Program, you'll be joining a vibrant community of like-minded researchers, practitioners, and industry experts who share a common goal: advancing orthodontics for the benefit of patients worldwide. The IOF offers 3 types of research grants for 2023: the Young Research Grant, the Elite Research Grant, and the Clinical Research Grant. The Young Research Grant supports graduate students, residents, attending physicians, and professionals in orthodontics and related fields. It encourages the development of teaching, patient care, and research activities. The Elite Research Grant targets assistant professors, associate professors, and professionals with orthodontic qualifications. It promotes outstanding research, emphasizing originality of the hypothesis and clinical relevance. The clinical research is a dynamic and collaborative field specifically designed for clinical research teams from multiple centers and universities who have demonstrated exceptional professionalism and achieved remarkable milestones in their respective areas of expertise. The 3 grants empower researchers in orthodontics and related fields, enabling them to pursue innovative ideas and drive advancements in clinical research, translational research, and patient care. The evaluation of all the proposals will be carried out by the IOF Science Committee. They will conduct a comprehensive assessment of each application, taking into account the scientific merit of the research proposal, the feasibility of the project, and the potential impact it may have on the field of orthodontics. Applications are open on June 16, 2023, and will be closed on October 31, 2023. The announcement of the selected proposals is planned to be made in spring of 2024. To apply for an IOF research grant, please visit our website. Before you begin, please read the guidelines carefully to ensure that your proposal meets our criteria. When you are ready to apply, look for an Apply button and take the time to thoroughly read and understand all the terms and conditions provided, which will direct you to an application form. Fill in the required information in the provided fields of the application form as instructed. We encourage all applicants to think outside the box and bring forward innovative research projects that have the potential to revolutionize the field of orthodontics. At IOF Foundation, we highly value research that not only upholds scientific rigor, but also offers practical applications for orthodontic treatment. If you have any questions, you can contact us via email at grants@iofglobal.org. Our team is here to assist you throughout the application process and provide any necessary clarification. You can also find more information by visiting our website. Stay connected with us on our social media platforms. We regularly update these platforms with exciting content, valuable resources, and important updates. Follow us today to stay informed and engaged. Thank you for considering an IOF research grant. We look forward to receiving your proposals and supporting innovative research projects that advance orthodontic education and improve patient outcomes.
So again, thank you. One thing I want to stress again one more time. For young people especially, if you're interested, don't get intimidated, don't get discouraged. Take the step, submit the grant, but don't use that GP chat thing, whatever. Submit your own idea as a grant. It's our job to help you, to improve your science, to get the grant, and to help your career, okay? So just do it, and we'll help you as we go along the way. Thank you. So next thing is the rising star. They asked me to present my talk first because 20 years ago, I was a rising star. It's just a little bit too late, sometimes it's too late. But I promise you, even today, I'm not talking about clinical work. I will not bore you with basic science. I just tell you that the journey of mine is a very fun journey, how I started and how I go from patient face up to the space and come back on patient care. It's a wonderful journey. So it's a story, just sit back and relax. I won't bore you with any hardcore science. So whenever I present my research, first thing I will show is the slide or the clinical patients that we care the most. There are a lot of things we can do besides appliance, biologically, and every different aspect. And this is our social responsibility, really, just like big corporate, successful company. A successful orthodontist It's our responsibility to take care of these patients, right? This shows a patient that I've seen long time ago, 1994, 95, since I first entered UCLA after Dr. Nam rejected me at West Virginia. So there's a patient has a cranial synostosis. What that means is that patients have overgrowth of their bone when they were growing. So when we look at patients, what can we do for them? And also, in addition to that, the bone occurs fast at this early stage. Can we isolate the molecules or the factors that induce this overgrowth bone and apply it on patients such as a cleft lip and palate, right? So at that time, we used a method called the dt-PCR, and we isolated comparing a normal suture and a fusing suture, screening through millions of genes, year work, we'll isolate one molecule called NEL1. And what is NEL1? NEL1 is a molecule, actually. It's very interesting when you look at the figures. Do you see that at the top, like a suction cup? What it means, it binds the molecules. That means this thing is doing something actively, right? Because it binds, it interacts. So we isolate the molecule, we put it back into animal model as overexpression. We put more of the genes into the rodent. What happened? We were successfully to recreate the phenotype, cranial synostosis. This is one of the very first models that we will be able to recreate. You can see that here you can see that the overgrowth bone here, right? It's very interesting scientifically. And what if we took it out? Took us a while to knock down the genes. And what you observe here is very interesting. When you overexpress, you have this cranial synostosis patients again. When you downregulate, what you observe strikingly, what happened to the mandible? It becomes microsomia and the missing of calvaria. So this gene we know when you too much or too little create craniofacial problems. And here, and after that, over the decades, we have a dedicated research team, primarily orthodontic residents and someone else. and they are all, and she's from West China University, Peking University, Tsinghua University. We dissect the pathway, and Monica actually found a novel receptor. It's very interesting. One day, hopefully, we can tell you the fun story. But it doesn't matter. If it doesn't do anything clinically, what's the point? It's like you watch Olympic gymnastics. People flip and flop. Does it affect your life? Not really, right? Yes, it gives you promotion, but so what? So we apply this molecule, we re-engineer the molecule to express in gene therapy and in protein form, and we increase the mineralizations. You see the calvario defect got increased. And here's the maxilla. We can regrade the bone in RPE. So one day, hopefully, Dr. Peter Nam will be able to use this technology to regrow bone. And we go to larger animals, all the way from sheep to primate. We're able to regenerate bones even with the vertebral body area. So you can see the fusion bone. This is the FDA analysis that we have done. We are the first group actually in orthodontics that published 3D FDA analysis. So with all being done, we know that this molecule can regenerate bone, right? During the wound healing and early growth. But at the same time, another group find actually this molecule is associated with osteoporosis. I'm sorry. Why is it? I mean, let me see. Okay. So we went back and re-examined the older mice now. Mice 18 months old, similar to, no offense, Dr. Nanda, around 70, 80 years old. So what we observe is that you can see the lighting, that means the bone formations and bone growth. You see this deficiency almost reflect the same thing as human, right? Why is it important? Because osteoporosis affects 1 of every 3 women over age 50, affects 1 of every 5 men over age, maybe 1 of 5 men, same thing, and contribute to 1.5 fracture. This fracture, usually when you get old, it lead to mortality. I think we all have relatives who fell and broke, unfortunately, their hip or their bone and went to hospital and never came back. So what we do is that can we use this protein a step further to use as a drug? So I worked with Dr. Benjamin Wu, and I worked with and his engineering chair at that time, and this is one of the famous fellow that we have. How we can make this molecule to be able to inject into blood and make it last longer, because a protein lasts very short time. So how can we make it last longer? So they spend a lot of effort trying to chemically bond, label the molecule so it can be protected from enzyme in blood, so it can last longer. And what's the effect of that? Actually, let me show you. The effect is that when we inject the molecule into bloodstream every 7 days, because you cannot inject every day, it's not practical. Every 7 days actually is quite practical. We're able to regenerate bone and prevent osteoporosis. And this is the FDA analysis again, when it shows the dynamic one. This shows the regular porotic bone. The different color, when you go to red, means bone is weakened, right? You can see the bone is sound and healthy when we put the molecule in every 7 days. This is pretty a major advance in what we know. And one interesting thing is that even for patients with, as you know, porosis have periodontal bone resorptions. When you put this molecule back, we're able to prevent the bone loss periodontally. So it doesn't just have an effect, in the skeletal aspect, has an effect actually dentally. So this journey took over 10 years. Many people, we found discovery to have models, we deliver it, and then we're able to use it systemically. In 2014, the NASA research director came and visit us, and we talk about research. It was quite intimidating to talk to present to them while in front of all the Nobel Prize laureate and candidate. The reason they have a problem, bone loss under microgravity. I think now China is launching its own space station, and the problem is that on Earth, you have 0.5% bone loss. What happens in space? You have 36-fold more. That means if you spend in space of 1 year without exercise, it's like 36 years old aging. Imagine the astronaut flying from Mars to back and forth. What happens to their bone? As a result, they have to exercise 3 to 4 hours a day, precious time on the space station, just to maintain their bone mass, right? So in 2014 and 15, we formally collaborated with NASA on their first live return R5 projects. And this we share as a news release. And this is the ambulance that we have done. So one of our residents designed this thing. This ambulance, every mission got an ambulance. This one represents a mouse study. So we're going to have a mouse in that ambulance. Do you see where's the mouse? The continent. Fun. Residents are always more innovative than we are. So again, we work with NASA team, BioServe team. Why is this important? Because again, not just the space. On Earth, microgravity reflects an extreme condition of bone loss. So it can benefit people on Earth too who have severe bone loss. And we work with the 12 core team. And you believe not, even FedEx does have a space team that transport professionally things between Kennedy Station and the research center. And of course, AOF is up there. I have to say this, when I first started this project, I couldn't get any funding from NIH because I have no connection, right? I'm not established. Even though my method works compared to other researchers who have $1 million grants, they won't fund me, except AOF. I was lucky. That was the very first year AOF established. I got the money. I took out a discarded laptop, Mac 2, Classic 2, from the staircase. I took it out, and I used it to my first grant. It was a horrible grant, but AOF funded me because they have trust in me. And that's how we became a huge project. The mission cost $140 million. It's a big project. Why is it unique? Why is it so exciting? First of all, it's the first time we'll be able to study a long period in space. Second thing, this is going to be a live return mission. Okay, it's never done before, and it's the first time for the US mission. And it's the first time we do a drug trial. And this is a team that we went and met with the NASA team people, talk about how to do it, and see that little thing here. That thing costs millions of dollars, as I show here. It's a space hub specifically designed for rodents so they can stay in space and station. And don't look down this thing. It take away all the defecation, urination under microgravity. Can you imagine? how you deal with those typical things in space. Again, this is a team. We have people, great people here. We have people, all orthodontics residents, faculty, residents, researchers, residents. So actually, it's a project, first project in dentistry with NASA, and done primarily led by orthodontic residents. We can do a lot more than we think. So as an aim one, you have to understand, when you work with ISS, everything on the space is going to be 10 times more difficult than on Earth, right? And how do you do this whole thing? Everything got to be practiced again and again in simulation on Earth. But the important part is that even though this $140 million project, the astronaut don't have much time for it. So we cannot inject every 7 days. We have to inject every 2 weeks, which is beyond the boundaries of what people can do in any type of growth factors in general. So again, we went back to Ben Wu. Unfortunately, he's not right here. Oh, he's here. So Ben, you know what you have done. So he made a very smart missile biologically. It's a bone-seeking delivery that he used the BP, injected PG as the guided missile target, one into a molecule, and you attack right on homing onto the bone surface. It sits there, and when bone get resorbed, it get released, right? And this is the data we have. 14-day injections now. We're able to recreate bone formation here. That's signified a big success, because now we are able to go to space. This after 3-4 years, the whole team work, and Monica, and they all involved greatly in this work. So this is how space work. 40 mice to the space. They fly up with the Dragonfly. It's the first SpaceX, actually, that went back to Earth, autopilot. And they go to space. 20 will stay for 9 weeks and come back. And 20 will come back to Earth. At the same time, at the Kennedy Space Station, the same amount of mice are being kept in a simulated environment, like simulating launching every single thing, and just to make sure we get an Earth. And these are 2 astronauts that we are very lucky to have. And Peggy Wilson, she's the lead astronaut, and she's been staying in space on the station for the longest. And this project is very dear to her. Think about, she was on space for over 2 years. Okay, imagine the amount of bone loss she suffered. So this is a very dear project to them. And this is the launching, and luckily, I have the opportunity to take my daughter as a VIP to the launching, to the Kennedy Station, and she's a star with the Elon Musk show, because she's the only one wearing the spacesuit. And this is our team. Here we brought our family, and it's my mother-in-law, and I'm thank God finally she's proud of me after all those years. And this just shows you, after launching, that's how we observe how the mice are alive and well. That's how they transport the mice to astronaut. On the micrograph, you can imagine what they have to do to old acrobat try to move this up, and that's how they work. So it's not as simple as on Earth, just use a pipette and do things. Everything got to be extra careful, protected. This is the timeline we've done. It was very interesting that actually 17, we launched on June 3rd. We received the first batch on July 4th, a celebration for the US holiday. It came back on the space pod, and then they ship it right back to UCLA. Okay, so again, this operation is very successful. We got 100% survival rate. They're alive and well. Last time didn't work well, unfortunately, and we have a large group samples. We're able to study what's going on, not just for our drugs, but in general, everything. We have a 4 team. Dr. Ben Wu, again, is a major team for the bioengineering. Dr. Wen Yanxi, Forsyth. Dr. Su is my wife, by the way. So we work together, and she's the PI. So this is what we have done. Over 20 postdoc graduates. Everyone worked day and night for this project. Okay, and 3 groups. Our bone group and the microbiome group by Wen Yanxi. The Ting she knows very well, and muscle group, because there's the 3 major things we should investigate. This is some of the data we have done. It's all officially okay by NASA, so we can release it without being arrested. So here is the data we get. It's interesting. Just look at the green and the blue. Green is the one with injection. Blue is the one without injection in space. So you can see the amount of bone loss we have. And the blue one and the green one shows you how we can reverse the bone loss under extreme gravity. And when we came back to Earth, we did the micro CT at around 10 micron, right? And we're able to compare the bone difference. And yes, we see the effect of the drugs. So this worked very well. Interestingly, this is something that's interesting we found. When you look at the histology, this is on Earth, typical bone marrow. When you went to space, what happened? Your bone marrow turned into fatty cells. So even though people don't observe, people don't notice that, but this became a problem over the long term. What happened to the astronaut, right? 10 years down the road. And interesting, we found that actually we're able to prevent it with the drug. And this is a life return with the same phenomenon. Actually, we're able to reverse that, and it's very interesting. I won't bore you with detail. And look at stem cell. When you fly space, you lost your stem cell. That's not reversible. So that is the issue we have seen. And then we tested with different type method to verify, yes, under microgravity, you lose stem cell. Long-term effect, no one knows. Okay, it should be under investigation in the future, especially for the astronauts' health. So again, we have shown that one works. That's great. And we are able to prevent bone loss. One thing interesting I want to show you all here. On this side and versus that side, that's a young mice. They're very young. They're like 20 years old at the back of the room. They knew that. When they go to space, what they do? They enjoy it. They fly. It's like they're playing with the microgravity. And these people of our age, what happened to them? They climb to the stage. They're scared. They don't move around. Young people, older people, younger faculty, older faculty. That's what we should know. That's why we should encourage the idea, right? That always make us think about, we should always behave like the young faculty and learn from them. So this is something I learned philosophically. And again, we do the large animal back on Earth. We're able to create the same thing in sheep model. So right now, this model, finally we got approved for clinical trial, and they are starting the first trial right before the COVID, I forgot. Okay, here is a complete circle. Sounds very fun, same time. How many years we've done? Over 25 years of all the people. Clinical problem we identify, study etiology. We learn it from the animal model, what happens to it, right? And then we go to the study the mechanism, that Monica's expertise especially. And then we have translational study to show what we can do with the molecule. And we apply clinically. We are at the stage of approval, first human trial. A study that would take another 5-10 years before it go on clinical trial. So take the whole career, 100 people to do the study. And this is what new drugs, and this is what we do as clinicians in academics in the long run. Nothing fast, nothing shortcut, but lasts a long meaning. And this is other people have done, the group. This is the muscle group. What they have shown is that for muscle atrophy, they identify the molecules, the mechanisms specified because of the atrophy. Through this, they are able to look for new drugs. So astronauts or people in the future can take the new drug and to prevent their muscle atrophy. And this is the fun part. This is the microbiome group by William Shi's group. What they found actually, the microbiome change on the microgravity. And those bacteria relates to bone loss. What that tells you is that perhaps we don't need a new drug completely. You can basically inoculate yourself with some bacteria that you don't want to take, but that helps you. So this is what happened. And finally, I want to show this, summarize. This is my wife, by the way.
To turn into bone cells and to grow bone. So it's critical for normal bone and to a certain extent cartilage development.
Orthodontic team.
The aim of this research is to test if NEL1 can successfully grow bone under extreme microgravity conditions so that
Fiona, our graduate, she's from West China, I think, Dr. Zhao's student.
We're going to launch up 40 rodents, treat the rodents with NEL1 systemically, and see if that will prevent microgravity-induced bone loss. So around 1886, with work from Dr.
This is a video of my resident filming at the restricted area in NASA.
There's a
SpaceX.
condition that little babies get called craniosynostoses, and that's a condition mission where in areas of active bone formation of this normal bone but prematurely formed,
Not just our
there's really
study,
high
what we
expression
have
of
done
gene
become the
called
standard
NEL1.
SOP for NASA
This
for the
gene
future
also
studies.
is
So the impact, not just on our
important
work,
for
is
preventing
related groundwork for all the
osteoporosis.
future NASA
Huge
studies.
problem, not just on Earth, but also in space.
Again, our residents, our faculty, all in Ortho.
I feel we're doing our research on Earth and we're lucky enough to be part of this that the other people have pioneered to make this possible.
Before the flight
Everything from
preparation,
Space
this is the
Act
flight.
To NASA Ames and all the tremendous equipment involved and the space station, all those missions to build the space
And this is the
station
autopilot for Tesla,
finally
fly right
brought
back to
this
the garage,
fruition
basically.
and made this
It's
possible.
very exciting
So
to see.
I think we're not really
This is the
pioneers
actual
in
image
that-
how our
that
animal being
sense at all,
transported.
but we're really just
Right here.
be able to take advantage of all the pioneering that other people have done.
40 mice.
And
First
then
time in
the other
history.
Tremendous exciting thing about this mission is that this is the first time
And
we're
we
going to
came
be
back to
doing
Earth
live
alive.
return. The rodent research hardware system that NASA Ames designed has to be rigorous enough to withstand not only launch in space, but for the mice to come back down. And so I think this is a tremendous achievement on their part to be able to think about all those things and have the rodents happy surviving in space and to make these experiments possible.
The FedEx space, actually,
have
they have
that
two
space
drivers,
space
simulation
flight,
flight.
but
They
also,
drove without stop all the way from
flights.
Kennedy Station to
Study
UCLA
out
at the fastest time they can get,
that
without any
really
rest.
Took us through step by step what needed to be done.
This is all our
to
dental students
make
and
this
our
mission
orthodontic
a
residents.
success. We're in our wildest dreams. We thought we would be working with cases and
Is Monica, you there?
experiments
Are you there too?
next week. And so we just feel tremendously privileged and grateful for all the pioneers ahead of us that really made this opportunity possible.
So to summarize what we've done, but we are far more productive. Two pair top scientists got married in my lab. Monica and her husband, the genius from Tsinghua University, and the pair, two residents. And people ask them, what's my secret? I tell them that lock them down in pairs in the lab 24/7. They'll get together. Dr. Chang remembered that, okay? Now they need to have babies, okay? So this is all the people we have from our lab, the past people that work with us. It's a big team, it's a group effort. And this, when we were invited to go to the White House, Eisenhower building to present next to the Space Security Council. So talk about our experience, and my wife, of course, is always the spokesperson for us, but this is how she took pictures. And this is the office here. It is the National Space Security Office. It's interesting. So she went to the deepest part of the White House. Again, people we have to appreciate, funding we appreciate. Again, AOF. Without AOF, we could have never done that. That's the spirit of AOF. That's why we're doing our IOF. We want to give opportunity to everyone, any interested young scientist in the world to have this opportunity, not just to look into the mouth, but look up. There's a lot more things up there we can learn, right? And again, this final thing I want to show you. This is the mission. I want to support AOF always as my talk. And IOF as the future. I want to support people in the future. And thanks to AOF and IOF people for supporting this thing. Exactly 30 minutes. Thank you very much.